The energy is because I finally, for the first time, got COVID. I'm recovering now, but it's it kind of threw me. And I got an American variant, which was much worse. I happen to. So, thank you, Christina, for that very kind introduction. Now, I've made my talk very much also aimed at patients and families. I presented this at HDSA because I think this is important for patients and families as well to understand. And so, after Dieter's beautiful talk, where he mentioned early intervention, we're going to go to a clinical talk where that's the aim. Okay, so what am I going to talk about? So the questions addressed in this talk is, quickly, what is clinical research? What is a medical staging system? Why do we need new? And what about existing methods? And who created it? What is it? How can I be staged? Do I need an MRI? Frequent questions. Does this change what we knew about HD? And how does this impact clinical trials now? And Amy already presented from PTC that now the HD-ISS is being used in trials. They're doing trials in HD-ISS stage 2, and many others are in development. So what is clinical research? So basically, clinical research involves human beings. It's studies performed in humans to advance medical knowledge. And we have amazing preclinical models, but the disease is Huntington's disease in humans. And clinical research is pivotal to us trying to develop our understanding of the disease. So for a successful study, you need to accurately select the correct participants. That may sound very simple, but I can tell you in so many fields and so many areas that doesn't happen. And you must be able to distinguish healthy people from controls. So what is a medical staging system? Now, I've got the example here, and I mentioned cancer in the clinical trial session this morning. Cancer staging system has revolutionized cancer trials. They can compare across studies. Everyone knows about the staging system. Cancers are all using the same staging system. So the stages divide people into groups with similar characteristics, and each group has a common problem, and a therapeutic is aimed at that common problem. So you have therapeutics for stages. And you need to understand which characteristics best define your group. So, cancer. So here, as many of you will know, stage 0 is when there are some cancerous cells. And it's the staging system based on size and spread. Stage 1 is a larger tumor, but it's still very localized. Stage 2 may have spread to a lymph node or nearby. Stage 3 is slightly more widespread, again, size and spread. And stage 4 is widespread metastatic disease. And so all the cancers use that staging system, and you can do cross comparisons across trials. Now, the European Medicines Agency and the FDA and all the other regulators want us to have clear and accepted staging systems that cover the totality of a disease, like they have for cancer. So why do we need this in Huntington's disease? What about existing methods? So, before the discovery of the gene, researchers still had to be sure who did or who did not have the disease. And I had a long and very interesting discussion with Anne Young recently at HDF about this. So the diagnosis had to be made when signs and symptoms were absolutely unmistakable. And this led to a diagnosis on clinical features based on motor diagnosis. So why do we need new? Because if you rely on clinical motor diagnosis, The DCL4, diagnostic confidence level, is very subjective between people. So relying on this for research is difficult because it's a subjective measure. And words which we use a lot of disease onset, manifest, pre-manifest, prodromal, pre-A, pre-B. We all have lots of different descriptions, but they all actually are slightly different, and some people may use them differently. So we need to have precise definitions, and we need it to be able to compare research results, and that's how the cancer field went forward, because they were able to directly compare trial after trial. We know that motor diagnosis occurs relatively late in the lifespan of a person with HD. And we know that other signs and symptoms, and patients and families have been telling us this for a long time, occur, and underlying biological changes exist, as colleagues have presented this morning and yesterday, much earlier. It's difficult currently to include trials with participants earlier in this disease state. And the regulators wanted us to have a staging system that would allow us to have the regulatory framework to do this. And the reason for this is because we want to do earlier stage clinical trials, as this may be the best time to have the biggest impact. And Dieter finished his talk by saying we need to do early intervention in C9, and this is the case for really all neurodegenerative diseases, and very much so in Huntington's disease. So who created it? It was created in the Regulatory Science Working Group, which I chaired from the Critical Path Institute and the HD Regulatory Science Group, and it was made up of HD researchers from academia. It was made up of clinician scientists. It was made up of industry partners and from pharma and biotech. And we examined all the scientific evidence to reach consensus conclusions. And we also had critical input along the way from the patients and families. We met with HD COPE early on. We got feedback from HDSA, we got feedback from EHA, we got feedback from HDO, and we also got feedback from the regulators. We published it this year after external peer review in the Lancet Neurology, and I'm going to give you a whistle-stop Porsche tour of the HD-ISS. So how did we learn all this about Huntington's disease? Well, we learned because of every single patient and family member who is in this audience or who may be looked after by people in this audience because of their participation in studies. We had the Enroll HD data, 20,000 individuals. We had Image HD from Nelly. We had Predict HD from Jane and colleagues. We had Track HD from myself and the team in Vancouver, Paris, and Leiden. And we had Cohort from HSG. So we have to say thank you to all the research participants who gave their time, because without this, we wouldn't have had the HD staging system. So what is it? It defines HD on the basis of the genetic test. It divides Huntington's disease into 4 stages: stage 0, stage 1, stage 2, and stage 3, which covers the full lifespan of a person with Huntington's disease. And the people in each stage share common predefined characteristics. So, why is it important? It's important for easy communication, so everyone can compare trials in HD-ISS stage 3, HD-ISS stage 2, because everyone speaks a common language. And this is really how we need to work with the regulators and all the authorities, because we will have better trials. It was developed to provide a universal use in clinical research, so we all speak the same language. It was an unambiguous definition of who is a person with HD that is standard across geography and allows clear communication. And we look at the entire lifespan of a person with Huntington's disease. And it does enable us to do clinical trials much earlier in the disease course. So how does it work? Huntington's disease is defined as the presence of a CAG repeat expansion of 40 or more in the Huntington gene. Some people with 36 to 39, and Darren spoke about this yesterday, and Michael presented from his lab. Galen presented some work really highlighting this is the reduced penetrance range, and some people won't go on to develop the disease. So they're only included in the definition of Huntington's disease if they have additional evidence such as biomarkers of pathogenesis or clinical signs and symptoms. So what is it? So here is stage 0. You carry the gene. And actually, the patients and family communities very much like the idea of stage 0. There is nothing to find except you carry the gene. Stage 1 is a biomarker of pathogenesis. And these gates are landmarks. So we have 2 landmarks for each stage. Stage 2 is clinical signs and symptoms. And stage 3 is functional change. Now, we know that everyone with 40 or more repeats will go through this stage and this course. Now, these dotted lines represent different paths. So, dependent on your genetic modifier burden or your genetic modifier in terms of good genetic modifiers, poorer genetic modifiers, environmental effects, many different things that affect progression. Some people may go slower, some people may go faster, but everyone goes in this direction. So, how did we come up with the stage landmarks? We had to do this in a very objective way. So, stage 1 was a biomarker of pathogenesis. Stage 2 was a clinical sign and symptom, and stage 3 was a functional change. And we did a literature review of everything that had ever been measured in Huntington's disease, and there was a lot of data. So there was actually nearly 3,000 potential landmarks. So because there's loads of studies in Huntington's disease, which is good. The literature review had to be data-driven and objective. We couldn't just choose our favorite thing. We wanted to have very strict criteria. The studies had to have more than 100 participants. They also had to have longitudinal data. They also needed to have prognostic value. And they needed to have prognostic or predictive value in 2 models or in 1 model that included 2 data sets. And we came up with the landmarks, and this was the process we went through as the committee, and we came up with, there were here 27 that were prognostic in models, and we had 16 strong candidates from all the clinical data, and 6 were chosen. They're those gates. And the landmarks were nominated and voted on through an NIH consensus process. And actually, interestingly, in the voting, there was unanimous voting for each of the landmarks that came up. So, Huntington's disease stage 0. Genetic definition, just the presence of the gene, nothing else. Biomarker of pathogenesis, the 2 landmarks and gates were caudate volume and putamen volume. Clinical sign and symptom, actually, total motor score performed very well. And symbol digit modality test, a cognitive task. And functional change was independent scale and total functional capacity. So many scales that we've had for a long time and have a lot of important data really performed well in terms of the landmarks. So how did we determine the cutoffs? So Jeff Long did a huge amount of work with this, and we were lucky through all the studies to have large numbers of control data, because you have to develop the landmarks using control data with cutoffs. And we modeled clinical data from non-HD people, similar to how the WHO growth curves are modeled. So those of you who have kids, these are very common in pediatric practice, and the district nurse always looks at this and says, yeah, the baby weighs fine. So, baby of 10 months old, the 5th percentile, which is the lowest, they would weigh about 7 kilograms. But the 50th percentile is like 8.5 kilograms, and that's modeled on normative data. So we developed cutoffs like the WHO models for growth, and there are many different examples of this. So we picked a cutoff in the controls, which based on the control data was an outlier. So we picked a cutoff for what we described as an outlier based on the control data. So it was scoring beyond the 5th percentile. So if, for example, your child is 10 months old and they're below the 5th percentile, then they are actually not gaining weight, and it's an outlier. So, an example. CAG 40, the caudate volume is below the 5th percentile for age, makes them stage 1. So it's a very objective way of measuring this. So this is it in a nutshell. Stage 0, stage 1, biomarker of pathogenesis, stage 2, clinical sign or symptom, stage 3, functional change. And stage 3 is divided into mild, moderate, and severe based on activities of daily living. And interestingly, this maps quite closely with the TFC as well and the independent scale. So ongoing work is mapping that. So this is an example of someone with CAG 42, the commonest CAG repeat. This is the probability of being in a stage. This is age. So someone is in stage 0 for quite a long time, and obviously it's that winding path. Some people are in stage 0 for longer, and some people are in stage 0 for less time. They will then go into stage 1, then stage 2, and then stage 3. The imaging, putamen and caudate volume, is pretty linear, and this is data with people with CAG 42. The TMS and the SDMT and the TFC and independent scale are very static, and then they start going down as you reach that stage. And Jeff, and in our paper, we've modeled this for every CAG from 40 to 50, the commonest CAGs that we look after. So, what's the impact of the HD-ISS for a person with Huntington's disease? Do I need to get an MRI? And this is a really common question. MRI is usually not needed. You can estimate your stage using statistical modeling. And we have a stage calculator with a website that you can put data into and get a staging. We can use clinical measures to deduce the HD-ISS stage because the stages always go in order. These inexact methods are okay because all the current clinical trials are focused on HD-ISS stage 2 and stage 3. Sometimes MRI will be needed for confirmation and to learn precisely about a person's actual HD stage, but MRI is done very, very commonly in clinical trials now. I think nearly every clinical trial is including MRI. So what's the impact? Does it change what we know about Huntington's disease? It doesn't change what we know about Huntington's disease. Historically, HD was perceived as a condition that appears decades after birth. But we know that clinical signs and symptoms occur prior to clinical motor diagnosis. And colleagues in this audience have been doing also a lot of work at looking at what may be happening in utero. And so the HD-ISS captures the totality of the disease. The clinical symptoms that we see are a consequence of changes in brain structure, which are themselves a consequence of changes in the function of cells. And the HD-ISS incorporates and categorizes this underlying biology, allowing clinical trials much earlier in the disease course. So, brain volumes, but not clinical measures, have similar linear trajectories. When you're planning a trial, the enroll database is very valuable because you can use the enroll data to stage potential participants before you enroll. So that's really critical, and the enroll database is so important. Trials, actually, the stages are quite potentially quite long, and the trials will benefit from HD-ISS staging and PIN enrichment. And the PIN, as many of you know, is CHE, age, SDMT, and TMS. And so you have PIN enrichment, and the HD-ISS staging system is a first-order classifier, but we need to do also enrichment methodologies like the PIN, and as Amy Lee presented it this morning, the PIN is an enrichment criteria in HD-ISS stage 2 in the PTC study. It improves your homogeneity and thus improves your power of your population and are critical for trial design. And this is the subject of a lot of ongoing work. So studies in HD-ISS stage 2 and stage 3 can be done now. They can be implemented immediately. We know how to enrich with the PIN and what to measure. Further work is ongoing for novel endpoints to increase power, reduce sample size in stage 2. And this is work in progress, and we're working on this in terms of endpoints for HD-ISS stage 2 with PIN enrichment, and it's watch this space. We've got some very interesting data and exciting data in the pipeline. So studies in stage 0 and stage 1. So I have presented on a number of occasions the HD young adult study, which we're doing, which is studying people decades before onset. We published the first paper in the Lancet Neurology about 2 years ago. It's studying very young HD gene carriers versus a large number of matched controls who are a mean age of predicted onset of 25 years before predicted onset. And we found in YAS that there truly was a stage 0, because in those there was a large group with nothing to find, which was great news at the time, and which we were very excited about. So that's stage 0. And trials in stage 1 are going to need enrichment and thinking about novel methodologies. Now, one of the things that we need to think about is clinical trials in stage 0 and stage 1 will be prevention trials. And we need further information on how we do enrichment and endpoint validation. But it's interesting to think that if we get a trial in stage 2 or 3 that meets its primary clinical endpoint, and shows that that primary clinical endpoint of function is associated with a biomarker that very clearly shows improvement, for example, and I'm just throwing this out there, I'm not being CSF neurofilament or brain imaging or the Huntington PET ligand or many other markers. The framework exists now for us to discuss with regulators, because we don't want to do 10-year stage 0 trials, is that if we show that we've reached a primary clinical endpoint in, for example, in HD-ISS stage 2 or 3, then designing stage 0 and 1, we potentially may be able to use the surrogate markers once we've validated them and linked to clinical benefits. So we will be doing trials in stage 0 and 1. So what's the impact? We've developed a biological definition of Huntington's disease that considers the spectrum of the known repeat expansion, and it's an evidence-based staging system. It has positive impacts on advancing therapeutic development. It unifies the research terminology for HD, which was much needed, and allows comparability across different trial populations. It has created a framework for the regulators to allow us to do intervention before motor diagnosis. And I can't really emphasize how important that is. We haven't been able to do that before. And now we have trials that are in the clinic before motor diagnosis. It allows the definition of study populations across the entire duration of the disease, including very early. And it truly paves the way for trials and treatments earlier in the progression of HD, with the hope to delay or prevent disease progression, which is why we're all here. So I want to say that this work would never have been possible without the vital contribution of every HD research participant who turned up for their track visit, who turned up for their cohort visit, who turned up for their enroll visit. This could not have been done without that. And also all the people who did the assessments, the doctors and nurses and trial coordinators. So the HD community's dedication to research has allowed the development of this staging system. So I often show this slide, but it's really because it's the way I think about it. I think of the world as one big laboratory. And the global community of HD families, scientists and clinicians, as in this audience, working together is our chance of achieving success. And I think the time is now. So I'd like to say thank you. I'd like to particularly thank Christina Sampao, Emily Gantman, Alexandra Mansbach and Jeff Long, who were a core team who did a huge amount of work to push this forward scientifically and experimentally. I'd like to thank other people who were in the group, who are in this audience, and particularly Chris Ross, Tiago, amazing contributions. I'd like to thank CHDI for having the vision to fund this, which I think will revolutionize HD trials. And again, I'd like to emphasize it wouldn't have been possible without the HD community's participation. So the time is, I hope, truly now, and I know this has been a really long day for you. So I'm happy, and thank you for listening, and I'm happy to take any questions.
So thank you, Sarah. I think it would need a very, very well-powered trial to show differences in the levels of energy of Sarah in this presentation and any other you might remember. So
no, this was Porsche.
This is Porsche. So any questions? We can one minute maybe.
Hello. Thank you. Very nice lecture. And I would ask a question about the new identification of biomarkers like neurofilament light, and how this will impact the scale of the HD-ISS. So meaning now, for example, for stage 1, you are using the volume of the striatum, and I was thinking now, if the NFL was discovered as a suitable biomarker, will it be included in this assessment as well?
Very good question. So NFL is very, very important. I'm very interested in it. Henrik did an amazing talk today. It's a big topic, both as a safety biomarker and as a marker of potential saving neurons. So the landmarks that we have are evidence-based and are fixed. But the question you have about CSF NFL is absolute. What we need is more data. We need data in hundreds of individuals, longitudinally in more than one study, because we need to have a very clear evidence base, and we need data and controls, because we need to get those normative cutoffs of what the change is, for example, change above the 5th percentile. So in the HD-ISS cohort, for example, cross-sectional, the work is ongoing to follow them up. We found it was about 47% of participants had a very subtle increase in CSF NFL. That was the only thing we found. 53% had CSF NFL the same as matched controls, no difference. So I think CSF NFL is very interesting as a very early marker of neurodegeneration. We need more data. I can see it being used as enrichment methodologies for stage 0 or 1. Once we get very good control data to get cutoffs, because you could envisage using pin enrichment, potentially neurofilament enrichment for stage 0 and 1 trials. And it's obviously a very interesting biomarker in terms of what Henrik presented with MS, nusinersen, tofersen. All those studies have shown that lowering it correlates with clinical improvement. And so, very, very good question, ongoing work.
Sorry, I think I'm next. So, Sarah, this was a really fantastic talk, and I think it's very important. Although, maybe I'm a bit hypersensitive regarding these issues, and to call something where there are no signs of a disease. So like Huntington's disease, stage 0. And when we do genetic counseling, I often tell the gene carriers that you are not sick, that you only carry the gene. And now, at least in this staging system, I tell them they have Huntington's disease stage 0. So were there any discussions regarding this framework and the naming this disease without disease signs?
It's a very important question. So the reason I mentioned HD-COPE, HDSA, HDYO, EHA was because that was exactly of that. This is for clinical research. This is not for your day-to-day clinic where you see subjects. This is for trials. This is for clinical research. That was why I started with a slide about what is clinical research. What we had overwhelming support from the patients and families was to have this stage 0. So for clinical research, every trial that you're recruited for now, you have to have a genetic test to get into it. It's just, that's what is our clinical research. All the staging system is doing is staging you. It's not for when you see people in the clinic and you say you have stage 0 and you have Huntington's disease. This is for research. So, and we had overwhelming support from the patients and families because they all want to do earlier phase trials. And we needed the regulatory framework to do that. So you carry the gene, nothing else to find. So it's for clinical research. But it's a good question. And obviously, it will impact patients and families, but it's really allowing us to do earlier phase trials because we don't want to be stuck in a regulatory framework to be doing just stage 3 trials forever. So I think but it's a good question, but this is for research. It's not for use. I'm not using this in my day-to-day Huntington's clinic. This is for recruiting for trials or any other study where you want to map biomarker data for clinical trials.
So let's have the last question there. Yes, the mic on end.
Professor, I'm Ali. I'm a family member of Huntington disease, and also I'm a neuroscience student from University of Palermo. You divided Huntington disease to 4 stages based on neurological symptoms. And I was wondering about the 0 stage, because I was living with Huntington patients. What is the cutoff between zero stage because
And stage 1?
no. What is the cut? When we divided psychological symptoms of these patients, because I was witnesses in the zero stage, they have some psychological symptoms. I can recognize a child with Huntington disease because some of them are very jealous, have very bad headaches. I was wondering, maybe zero stage can be divided to two phases. The second phases can be start of the psychological symptoms of these childs.
So it's a good question. We did look at every study that had measured psychological and psychiatric symptoms and psychiatric burden, and you're absolutely right. But there was none that could very, very clearly fit the consensus criteria as being very clearly and robustly associated in a way that was measurable with normative cutoffs for staging. You make a great point, though. So what we found in HD young adult study was very interesting. This was gene carriers mostly in their 20s, decades before onset, but we also had age-matched controls who were also in their 20s. And actually, and it was a great surprise to me, having looked after patients for over 25 years, is that we didn't find a difference in a huge battery of psychiatric symptomatology. The young people in their 20s who did not have the Huntington gene actually had more anxiety and depression than those who did. So you're absolutely right. We know there is psychological symptoms, but they just are too variable to go into a staging system for clinical trials. And also, a good thing is many of them are treatable with SSRIs and many of the medications we use. So a very good question, but I think it's something in terms of the staging system, we needed a kind of a very objective one that would not be variable. So, but a very good point.
Thank you, Sarah. This was excellent.